नमस्कार। आप सभी लोग अपने अपने घरों पर सकुशल होंगे, ऐसी प्रभु से कामना है तथा अनलॉक होने पे भी आप सभी लोग कोविड-19 से बचने के उपायों को प्रॉपर कर रहे होंगे। इसी कामना के साथ हम बात करते हैं अपने टॉपिक की। एज वी आर टॉकिंग अबाउट द मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस इन विच वी हैव टॉक अबाउट डिफरेंट experiments which are given by the different scientists. Then let's move to another topic, a new topic, that is the RNA world. Means generally we have to name that what is the first genetic material, DNA or the RNA. Means generally there are lots of things which can be asked by the different peoples, that what is the first genetic material. For that, according to different metallurgical or metabolism, especially by the translations and splicing, evolved around the RNA. Means generally all these processes, evidences which can be always helpful for the suggesting that RNA was the first genetic material which was discovered. And if we take about their evidences, especially to prove that the essential life process such as metabolism, translation, splicing, all these can be have because of evolving around the RNA. मीन all जितनी भी प्रोसेस हमारी लाइफ की हैं, चाहे वो ट्रांसलेशन की हो, स्प्लाइसिंग की हो, मेटाबॉलिज्म को, ये कहीं ना कहीं आरएनए के आसपास से ही मूव करती हैं. तो जिसकी वजह से हम कह सकते हैं कि आरएनए इज द फर्स्ट जेनेटिक मटेरियल. और वी कैन से some important biochemical reactions in the living system that are especially catalyzed or can be started with the help of RNA catalyst or not by the protein enzyme. Means especially different processes can be started because of the catalyst. RNA is found over there, or we can say DNA has evolved from the RNA, which can be chemical modification that made it more stable, because RNA being a catalyst was a रिएक्टिव एंड हेंस अनस्टेबल. मीन्स जनरली अगर हम बात करते हैं डीएनए की, तो डीएनए इवॉल्व फ्रॉम मीन्स डेवलप फ्रॉम द आरएनए, और ड्यू टू सम केमिकल मॉडिफिकेशंस इट कैन बिकम्स मोर स्टेबल इन कंपेयर टू आरएनए. एंड आरएनए इज अनस्टेबल बिकॉज ऑफ इट इज एक्ट्स लाइक ए कैटलिस्ट, सो इट इज रिएक्टिव, so it is unstable over there. So on the basis of this RNA, then generally there are three types of RNAs are found, that is messenger RNA, transfer RNA and rRNA, ribosomal RNA. We all have to know about that, especially about this, that this can be shows the different mechanisms in different processes over there. First, if we can talk about this, especially RNA, that messenger RNA, that can be always help to provide the template for the transcriptions, and transfer RNA, that being amino acids and reads the genetic codes for the different genetic materials. Or ribosomal RNA, we can say it can play a structural and the catalytic role during the translation processes. Or we can say all these three RNAs are needed to synthesize the protein in any cell. Means all the three cells, especially transfer RNA, messenger RNA and ribosomal RNA, all these three kinds of RNA is needed for the formation of the protein in any cell, right? Evenly we can say that the modification includes the double standard nature, complementary strands, resist the changes evolving in that particular conditions. After that, we have to know about the replications. What about the replication? Means we can say the replication, or we can say the replication of DNA is termed as the semi-conservative DNA replications. Means generally replication of the DNA is known as semi-conservative replication DNA, right? Especially if we can say that from the experiments or from the process of which is proposed by the Watson and Crick, right? The double helical structure, and the Watson and Crick original statement that he explained that it was not escaped out. Notice that the specific pairing we have postulated that immediately suggests a possible copying mechanism from the genetic material. Means they explain about that the DNA is a replication, or that is a semi-conservative nature through the semi-conservative method. Means this was the semi-conservative DNA replication. It was proposed by the Watson and Crick in 1953. According to him, he gave the specific things that the two strands would be separated and acts a template for the synthesis of a new complementary strand. उन्होंने कहा कि जो डीएनए की दो स्ट्रैंड्स होती हैं, दे विल बी सेपरेटेड, वो आपस में अलग हो जाती हैं और बिकम्स अ टेम्पलेट for the synthesis of the new strands, new complementary strands should be synthesized over there. After that application, each DNA molecule would have one parental and one newly synthesized strands are found over there. Means if we can say with the help of diagrams, suppose we can say this is a double helix structure over there, right? And after that the replication, there will be change over there, right? Means this strand is also separated from over there, this is also separated over there, and this is known as the template, right? Means it becomes the template one, and in the same time there is another, it will synthesize a new arm that is called the complementary DNA over there during the replication. I think everybody understand that this is the DNA which can be replicated over there, right? And it will change into template arms, as well as the new DNA will synthesize with the help of the complementary strands. So it can be found over there. So we can say that newly synthesized strands are found over there, and the basis of semi-conservative methods replication completely between the nitrogen bases. Generally, the bases is also between them, that is nitrogenous bases are over there. So with the help of this, the Watson and Crick can explain about this, that shows the DNA is a semi-conservative in nature. It is experimentally proved. Generally, the DNA replication semi-conservative becomes from Escherichia coli and later is higher organisms such as plants and humans. First of all, it's been proved with the help of the special process, right? Especially in the Escherichia coli, but later on it can also proved into the plants and animal genetic material over there. So Matthew Michelson and the Franklin Stahl, they performed an experiment to prove this DNA is a genetic material or DNA is a semi-conservative in nature in the laboratory with the help of its conditions, as you can see in the diagram over there, right? According to his experiments, generally they have used, he also shows that the conservative replications would leave intact an original DNA molecule and generate the completely new molecules, as well as this driving replication would produce two DNA molecules with sections, and both old and new are found over there. And semi-conservative is generally it will produces molecules with the both old and new, but each molecule would be the composed of the one old strand and one new strand. That's why it is known as semi-conservative in nature. You can see in the diagrams as well as in your books also, there are different methods through which we can capable to see. So if we have to directly comes on the experimental which can proposed or done by the Matthew Michelson and the Franklin Stahl in 1958. He started to grow, right? Escherichia coli in a medium that is called 15NH4Cl as only nitrogenous source of for many generations. Means generally it is the nitrogenous ammonium chloride over there. So it is, we can say that N15 is a heavy isotope of the nitrogen, and it got incorporated into new synthesized DNA and other nitrogen containing compounds over there. So these heavy DNA molecules could be distinguished from the normal DNA by the certification in a chemical that is called cesium chloride with the help of density gradient over there. After that, They then transport the cells into the medium with normal NH14 NH4Cl and took samples at various definite intervals as the cells multiplied ejectly DNA and remains the double stranded helix. The DNA sample were separated independently on the cesium chloride gradients to measure the DNA densities, and then the DNA was extracted from the culture first generation. After 20 minutes, that it transferred from the 15N to 14N medium and had a hybrid intermediate density means one strands of DNA 15N while the other strands of DNA, that is 14N, found over there, that can be extracted from the culture after that second generations. After 40 minutes was composed to equal amount of the hybrid DNA and the light DNA, which is found one DNA is molecule was N15 and N14, while the another DNA that was N14N and 14N. So they explain the semi-conservative method of DNA replication was over there. According to the similar experiment was also done by the Taylor and colleagues in a special plant, that is called Vasia faba or faba beans, using the radioactive thymidine, and same results were obtained in the earlier experiment in the DNA replication or that it can be always shows the DNA is semi-conservative in nature, as you can see in the diagram over there, right? That this is the parental DNA and he uses the N15 where after the templating it can be when it can transfer into the medium of 14N NH4Cl, it can be just developed into this nature, means one arm with N15 NaCl or N15 isotope nitrogen and the light isotope. Later on, after 40 minutes, it can be just transferred into the next generations where we get the such structures. Means generally you can found that after in the second generation the DNA molecule can changes into such conditions. You can easily understand with the help of this that this is the first and the later on this is the second one. After in the second generation, after 40 minutes in the culture medium, we will get the 15N and 14N two strands. Evenly there is a 14N, they are two one pairs, and again 14N and 15N is also called. So this can shows that DNA is a semi-conservative nature, means the one arms is always belongs to the parental one, that is 15N, it always found over there, and after the synthesizing evenly there is a one more arm is also N15 over here and 15 over here. So these are the parental arms, parental DNA, and the later on these are the hybrid one, so they can be developed the newly synthesized. So N15 is the heavier isotope, so it can be found over there, and N14 are lighter chain over there. So we can say, as I told you, that after extracting of DNA from the culture, the first generation after 20 minutes that can transfer from 15N to 14N medium and had hybrid intermediate density over there. So means one strand of DNA is N15 and second one is the N14. And DNA extracted from the culture after second generation, after 40 minutes we can say, was composed of the equal amount of this hybrid DNA of the light DNA. Means we can get the hybrid DNA and that is heavy and the light nitrogen base, evenly the light one, that is N14 and N14, and one molecule, or we can say the amount of molecules, DNA molecules, that is 15N and 41 and 14N and 14N is always equal to both ones. So we can say they are the semi-conservative in nature. So this is the mechanism of the semi-conservations of that particular structures in the DNA. We have to talk how this process, how the DNA replication will take place. There are different types of enzymes are participated over there. Different types of substances, different types of enzymes can be participated over there. Which one are those? You will have to also know about them. So first of all, we have to talk about the DNA replication over there. So we can say the process of replications have required different substances, right, or a different catalyst or enzymes over there. So we can say DNA replication take place in the S phase of the cell cycle. Means if we talk about the cell division in any cells, there is a different phase, and M phase, P phase and S phase, right. So the DNA replication will always take place during the S phase of the cell cycle or cell division, or we can say the main enzymes, that is DNA dependent DNA polymerase, right, as well as we can say it used to DNA template catalyst polymerizations of the deoxynucleotides, as well as the average rate of polymerization by these enzymes is approximately, that is 2000 base pair per second. When this enzyme, that DNA polymerase, can capable to polymerize approximately, that is 2000 base pair per second. Even we can say the polymerase has to catalyze the reaction with high degree and accuracy, because many mistakes during the replication would be results into the mutation. Means generally जो polymerase enzymes होते हैं, polymerase catalyst होते हैं, they can be very accuracy, because if there is any mistake, what will happen, then mutation will take place, means the sudden change can be are found over there. So this polymerization is very accuracy and accurate degree. Even we can say the DNA polymerization is an energy demanding process, means generally lots of energy can be consumed in this DNA polymerizations. So deoxyribonucleosides triphosphates serve as the dual purpose, means deoxyribonucleoside triphosphate, right, it can be shows the two process, दोनों तरीके से काम करती है, that it can act like a substrate, provide energy and for the polymerization. Means generally it can be act like a material also, it can also act like a substrate which can provide a large amount of energy for the polymerization reaction over there. Or we can say many additional enzymes are also required in the addition of DNA depending, that is DNA polymerase, such as helicase, topoisomerase, SSB and ligase. Means we have also known about their roles. What are their roles were? Even we can say the both strands of DNA get separated by activity of, that is called helicase. Means helicase enzyme can be separate them. Means the both helix can be separated because of the helicase. It can be always help to unwind the DNA. As you know that जो DNA होता है वो ट्विस्टिंग होता है, विंडिंग होता है, मीन्स एकदम घुमा हुआ होता है, तो उसको अनविंड करने का काम और उनको सेपरेट करने का काम कौन करता है? हेलिकेज एंजाइम करता है. ऑन द अदर हैंड दैट इज टोपोआइसोमेरेज, दिस एंजाइम कैन रिमूव द टेंशंस. मीन्स जब वो ट्विस्ट करता है, उस ट्विस्टिंग को थोड़ा सा अनकॉइल करते समय उसकी जो टेंशन है, जो खिंचाव है, उसको रोक के रखता है. इवनली द another, that is called SSB protein. Then SSB protein can always help for the stabilize the uncoiled DNA strand, so that cannot be coiled again. Means generally this SSB protein, after the unwinding with the help of topoisomerase, it can be capable to maintain this SSB protein. It can be make the stabilize that the it can be becomes an uncoiled. After the opening, it cannot be again going to be coiled one. So we can say the uncoiling DNA strands form a Y-shaped structure that is known as replication fork, right? So that replication fork can be found over there in that particular conditions. So with the help of these all things, this can be happen over there. Similarly, we can say the other different part that are replication in DNA strands occurs within the small opening of the DNA helix known as replication fork. Means I think you all have seen the structure of the DNA. This is known as replication fork over there, right? That is 5' to 3' and 3' to 5'. So there is the replication process is known as over there. So different enzymes can act over there, right? After we can say the DNA polymerase cannot initiate the process of replication on their own. Means this DNA polymerase enzyme directly don't initiate it, but it can be needed a primer to start the process DNA replication. Means it needs another substances कि जैसे हम आपसे कहें बेटा आप अपने आप नहीं पढ़ते होंगे, but you have to just given instruction by your teachers, by your parents, then you started to join your class, then you started to do your work. In the same way, the DNA polymerization cannot initiate itself, अपने आप नहीं होती है, it needed a primer टू स्टार्ट दिस प्रोसेस. आपने सुनी होगी दैट चेन रिएक्शन में जब तक एक इलेक्ट्रॉन बम्बार्डमेंट नहीं होता है तब तक चेन रिएक्शन स्टार्ट नहीं होती है, लेकिन आफ्टर बम्बार्डमेंट इलेक्ट्रॉन द रिएक्शन विल स्टार्ट. इन द सेम वे इन द डीएनए रिप्लिकेशन दिस डीएनए पॉलीमराइज डज नॉट डू इट्स रिप्लिकेटिंग प्रोसेस बट इट नीडेड अ प्राइमर फॉर द प्रोसेस. सो वी कैन से डीएनए डिपेंडेंट डीएनए पॉलीमरेज catalyze the polymerizations only in one direction, that is 5 prime to 3 prime, right? It can always creates you can just see in the diagram over there, right? It can always help to create the different processes over there. That DNA polymerase catalyze polymerizations only in one direction, that is 5 prime to 3 prime, right? So we can say that one stands, that is template stands 3 prime to 5 prime, right, and replicate in continuous. This is known as leading strands. You can just see especially in the diagram over there, right? Means we can talk that it can be always created from the 5 prime to 3 prime in the diagram, right, here to here. But after that this is the leading strands, leading one. So we can say especially 3 prime to 5 prime. While on the other strands, that is template, that is 5 prime to, you can just see in this diagram especially this is the 5 prime to 3 prime, right? So there is another primer can be also used over there. So we can say you must understand that the DNA depends DNA polymerase catalyze polymerization only in one direction, that is 5 prime to 3 prime, and it creates additional complications at the replication fork on one strands, that is template strands, that is 3 prime to 5 prime over there, right? And in the same way on the other frog, that is 5 prime to 3 prime over there. These are the leading strands, while the other we can say, right, means generally in the first one, in this condition, this is the leading one and this is the lagging one, means that 3 to 5 that is leading and 5 to 3 that is lagging strand. We can say that the discontinuous synthesized fragments, they are known as Okazaki fragments. You can see especially in this one, these are the Okazaki fragment here, these are the discontinuous parts are found over there like this, right? So these are known as Okazaki fragments, which can be later on rejoined over there like this one, right? So we can say that discontinuous synthesized fragments called Okazaki fragments are the later joined by the DNA ligase, means with the help of DNA enzymes, DNA ligase enzymes, these can be capable to join over there in the easily in that conditions. So I think you will all understand with the help of this one. So you all must need that you have to memorize all these things over there, that how these can be available to rejoining these conditions in that particular condition in the replication conditions. So you must know that during the replication, as I told you, this replication not done itself, it needed a different primers, and this polymerization can be always in one direction. We can say that is 5 prime to 3 prime, and it is because of the replicating fork can be developed over there in which the leading strand and the lagging strands, and there are some structures which are unjointed or discontinuous structure which can form the Okazaki fragments, and later on these Okazaki fragments can be joined with DNA with the help of the DNA ligase enzyme, and it can always helpful for the developmenting of the replication of DNA. Then we have to know also that origin of replication, where it can originated. We must know that DNA polymerase cannot initiate the process of the replication on their own, right? And also replication does not initiate randomly at the place in the DNA. So there is a definite region in the Escherichia coli DNA that where the replication originated. This region is termed as the origin of the replication. Means generally अपने आप दिस प्रोसेस नहीं हो पाती है, लेकिन इसके बावजूद स्पेशली इट कैन बी नीडेड सम स्पेशल प्रोसेसेस इन विच दे कैन बी कैपेबल टू replicate it, means it can be initiated to particular place where this process will start to replicate that particular thing, that is we can say it can be start to replicate it, and that place is known as the origin of replications. On the other, we can say due to this requirement, a piece of DNA if needed to be propagate during the recombinations, DNA proceeds and requires a vector. The vectors provide the origin of the replications over there. Means these are different. Means this origin of replication places needed different things also. It needs a piece of DNA, right, to be which can be recombinant over there, and it also requires a vector which can be do that process, and it can search for the origin of replication place over there. So this way the origin of replication or this replication process will develop into the DNA.